The aim of this procedure is to monitor how proteolysis of cyclin B governs anaphase onset and mitotic exit. This is accomplished by first seeding cyclin B SNAP reporter cells on microscopy slides. The second step is to label the chimeric cyclin B SNAP reporter molecule with the fluorescent substrate TMR*. Next, the image series are acquired at a microscopy station. The final step is to analyze cells and calculate TMR* fluorescence intensity over time, which reflects cyclin B proteolysis. Ultimately, immunofluorescence microscopy of living reporter cells reveals the kinetics of cyclin B proteolysis taking place during mitosis.
The main advantage of this technique over existing methods, like monitoring cyclin B GFP, is that cyclin B SNAP allows monitoring of cyclin B degradation rather than whole protein expression.
The SNAP reporter cells for this experiment are initially allowed to grow asynchronously in log phase for at least 48 hours. To begin the procedure for seeding, trypsinize the subconfluent SNAP reporter cells. For seeding of cells onto 8 well microscope chambers at a constant distribution across the entire surface of the chamber, centrifuge 10,000 cells and resuspend in 350 microliters of phenol red-free normal growth medium.